Okay, so now we want to talk about Chapter 3, which is all about organic molecules. We're particularly going to focus on some of these macromolecules. This is where we are on our study of biology. And this falls under what we would call biochemistry, or the study of the chemistry of living things, which is really centered on these organic molecules, these molecules that are carbon-based. Okay. Now, so these molecules, as we've seen, will always have a carbon structure with various things stuck on them. In particular, these functional groups. You'll have your carbon skeleton, and then these different, of course you'll have hydrogen stuck on, but then you'll see these typical, what are called functional groups. So we talked about the hydroxyl groups, the OHs, and while they are always found in alcohols, they're also found in some other kinds of molecules, and their main claim to fame is that they add some polarity to the molecule. That oxygen, again, is sort of hogging some of those electrons, and so it gives that part of the molecule a slight charge. We also see these carbonyl groups, which is basically a carbon double bonded to an oxygen, and here this R in each of these just represents the rest of the molecule. And you'll notice that sometimes the carbonyl group is sort of at the end of the molecule with the double bonded oxygen and the hydrogen, and sometimes it's in the middle of the molecule with more of the molecule to the right and the left. But either way, it's always this carbon double bonded to an oxygen. Don't worry about these terms. Just know that, again, this, because of the oxygen, this functional group gives that molecule some polarity. All right, the carboxyl group, which we have seen in our discussion so far, sort of has this chemical, whoops, go away, this chemical formula. But here's the arrangement here, and you'll recall that because of this OH group here, that hydrogen tends to break off, and so these are sometimes known as carboxylic acids, because that oxygen or that hydrogen will break off, and so this part of the molecule will have a negative charge, and you'll have that hydrogen, which in a solution will form some of this, which will decrease your pH, thus acidifying the solution. Okay, the amine groups, the NH2, they also give the molecule some polarity. We'll see that they are found in amino acids. That's a common place we find them. They, as is also pointed out here, tend to cause the solution to become more basic because they have the tendency of grabbing onto an extra hydrogen, thus taking that hydrogen out of solution, which will raise our pH, make it more basic. SH, or sulfhydryl groups, they, as we'll see when we study proteins and amino acids, you find them in some amino acids, and they give the amino acids the ability to bond to each other through what are called these disulfide bridges. So the sulfhydryl group on one amino acid and the other amino acid, they'll ditch their hydrogens, and then those two sulfurs will bond each other. That's what's known as this disulfide bond. Okay. All right, and then last but not least, the phosphate group, which is sometimes abbreviated as PO4, but really kind of looks, this one's kind of a funny way to look at it, but it often has this kind of look. Oops, I meant that to be an oxygen. And so what happens is it'll be bonded to one of the oxygens. there'll be an oxygen between the phosphorus and the rest of the molecule, and there'll be a couple other oxygens that tend to grab onto hydrogens. But again, we tend to abbreviate it this way. We'll see it adds some polarity to certain molecules, and we find it in some amino acids, and we'll see it's in what's called adenosine triphosphate, or ATP. And when we study certain biochemical pathways, we'll see that ATP is very important in transferring energy, moving energy around. Okay. All right, so these are the big four, or the four macromolecules that we're interested in biochemistry. Carbohydrates are basically sugars. Proteins, which, as we'll see, they have the fancy term polypeptides. Lipids, which come in various types. There are fats, oils, phospholipids, and also steroids and waxes. We won't worry about waxes too much, but we will talk about these others. And then the nucleic acids, RNA, it's an A, and DNA. Now, three of these molecules, in particular carbs, proteins, and nucleic acids, are what we would call true polymers. And that is, they are made up of basic units known as monomers to make up polymers. That is, you put a bunch of monomers together and you get a polymer. And these molecules can be tens to hundreds, thousands, and in the case of some nucleic acids, even millions of monomers long. They can be incredibly long, very large molecules. We'll see how lipids are a little different. They don't come as true polymers. Now, when we want to take these monomers and put them together into polymers, we have a particular reaction that we do. And of course, we are making polymers out of monomers. We're doing the act of polymerization. And this occurs via what's called dehydration synthesis, or a condensation reaction. And that is, we have these two simple sugars here. We're going to visit those in the next section. And what we're going to do is, to bond them together, we're going to move a hydroxyl from one and the hydrogen from the other. And you'll see the remaining oxygen is going to bond between them, and we give water off. Again, you might say, dehydrating the molecule. We're removing water from it, or water is condensing off of it. So to take these two monomers and put them together, we remove water in this dehydration synthesis reaction. If we want to take two monomers apart, we do the reverse. We put water in and split them apart, and this is known as hydrolysis or hydrolysis, where to lyse means to split, and we're splitting with some water. Okay, that's it for section 3.1.